Yeah, started. Any questions from yesterday? Okay, today, couple of lectures, I'm going to talk about some of the issues that crop up while we are building phylogenies, some of the complications, and how we should be interpreting some of these results. And then I'll get to frequently asked questions. So all through the workshop, many of you have been asking me questions regarding some analysis or how many markers to use and stuff like that. So we'll get to that today. Okay, so yesterday we discussed briefly gene trees versus species trees, right? And so this green, thick, whatever line or whatever you want to call it, is the actual species tree. And let's say we know what the species tree looks like, which is basically a phylogeny. And then those dashed line and thick line there inside, embedded within the species tree, are the gene phylogenies or gene genealogies, right? And one would expect to get the same exact gene tree, right? When you look at multiple genes, you should get the same gene tree, and it should be congruent with the species tree. And that kind of makes sense because genes are embedded within species, right? And every time there's a speciation event, there has to be gene divergence. And what are we doing? We don't know the species tree, we don't know the phylogeny, so we are building gene trees and inferring the species phylogeny from that, right? And we are assuming that the relationship between species in the gene tree is basically giving us the phylogeny, okay? So typically we expect multiple markers to give the same exact tree. Now yesterday we spoke of a complication where it does not, where the gene trees can be different from each other. What was that? Any thoughts? Forgotten already? And we talked about under such situation we should use a different approach of tree building. Species tree approach, right? So what was the issue there? Clearly the individual gene trees were different, but why?
Depending on the function of the gene, whether it is fast evolving or it is conserved.
Yeah.
Some function is retained in some species.
So that's the selection part you're talking about. We'll come to that later. But yesterday's lecture, we spoke about the species tree approach using a program called StarBeast. Huh? Deep coalescence. What is that? Yeah, so gene divergence prior to species divergence. But why does that happen? Incomplete linear sorting is another way of saying deep coalescence. But under what situation of speciation can we have this kind of a deep coalescent event?
Different evolutionary histories.
Yeah, but yeah, different evolutionary histories is again another way of saying deep coalescence. Remember we talked about two speciation events? There's the first speciation event here, the second speciation event, right? So what?
Close speciation.
Yeah, when these two speciation events are very close, right? So that's when you have this situation of getting two different topologies, because you have a lot of variation here, and basically the gene genealogies are telling us how those variants are related to each other, right? So under this condition where you have rapid radiation of multiple lineages from an ancestral species, individual gene trees can differ from each other. Okay, this is somewhat related to, so if this time period between the first speciation event and the second speciation event is long enough, then the alleles will sort themselves, right? And there'll be many synapomorphies that unite these two species. But when it is very short, there's just one or very few synapomorphies, and sometimes it can revert back to ancestral state and giving you a different tree. Or the alleles, if there are many alleles here in this population, they can sort themselves in a way that confounds species relationships, right? And that's when you can have different tree topologies or gene topologies. And one can do bootstrapping, and that will basically either show you something like this, which is, bootstrap support is less than 50, or if you ask the program, basically you'll realize that support for this topology or this topology, when you concatenate it, will be very low. So that's when things are going wrong, and then maybe you can also run an ILD test, incongruent length difference test, to figure out if the different genes have very different signals or not, right? And if that's what happens, if that's what turns out to be the case, then we use species tree approach, right? We use star beast. That's what we discussed yesterday. So this is one condition where genes can give you different topologies. And the other very common situation that which is related to deep coalescence, but this is speciation event that has occurred very recently. In fact, Chaitanya talked about it a little bit, right? So this is a condition where when the speciation event is very recent, species share lineages with each other, right? So they are polyphyletic, and then they go on to become paraphyletic, and then they become reciprocally monophyletic. Reciprocal, so both are monophyletic, that's why reciprocal monophyly. So if you're in this zone, individual genes can give you different topologies. So if the speciation event is very recent, the alleles have not sorted themselves, so this particular individual from species B is more closely related to an individual from species A. And for different genes, you can get different topologies. Whereas if the speciation event has occurred quite some time ago, then all the individuals in the two daughter species will cluster together. But further back in time over here, it will look more like that, right? Now, what's interesting here is we are using this term polyphyly, paraphyly, and monophyly. We have used these terms in another context. What was that? Huh? On day one.
You were studying the characters.
A little louder.
When you're studying the characters
Yes.
Human and
Yes, that was paraphyly, right? So, and then we looked at the vultures, it was polyphyly. So that is a different context, and that's why maybe these terms should not be used here, but so that is the context where there's discordance between taxonomy and molecular phylogeny. And to describe that discordance, we use these terms. Okay? Whereas here, I don't know, maybe in a way it's discordance because what we are calling A and B are not monophyletic. Right? So in a way, but it's a very different kind of discordance.
Ravi,
Huh.
in terms of a tree topology, like
Yeah.
strictly speaking, irrespective of the method we are employing for getting the tree topology, the terms would remain the same, right? In the like representation sense of it.
What terms are you talking about here?
Polyphily, paraphily, reciprocal,
Huh,
monophily.
yeah.
In terms of representation, the terms would not change.
Yeah, it
So
won't change.
interpretation, it may depend on what type of tree we have used. So to explain the discordance, it would depend on the type of tree which we have built, right?
Again, what do you mean by type of tree?
So whether it's a cladistic approach or whether it's a character state approach, depending upon that. So for example, in the old world, new world virtual case, we have physiological traits being the primary criteria for putting them into different trees.
Yeah,
Here we are using nuclear data, genetic data.
yeah.
So like in the representation sense of it, the terms would remain the same.
Yeah.
But for explaining the discordance, they are explaining different types of discordances.
Yeah. That's another way of looking at it. Yeah. So here it is more genealogical discordance. There it is, taxonomy versus phylogeny discordance, right? And this sort of makes sense. What happens is, early during the speciation, the daughter species, so this is where the barrier has arisen, daughter species will share a lot of alleles, right? So if you think about, say 2 alleles in a population, right? And these 2 alleles at a particular locus so let's say, there are 2 alleles A1 and A2 in a population. And so on, right? This is some population. And there is a barrier that appears. Now what has happened, you have 2 subpopulations, and slowly these 2 subpopulations begin to diverge. Now, when the barrier has just appeared, what you're seeing, you're seeing that they are sharing alleles. Right? That's basically polyphily. Because these individuals, which are technically in 2 different incipient species, are related to each other. Right? Now, give it enough time, one of the alleles will get fixed in one population and the other allele will get fixed in the other population. Right? This is a very simplified way of looking at it. So let's say, due to drift, A1 gets fixed here and A2 gets fixed here. This is reciprocal monophily. Right? So that is what is illustrated there. And if you are sampling these genes, if you are in this zone, you won't have monophily, and you would begin to question yourself whether these are 2 different species or not. They might show some morphological difference also, right? So typically, Avis was the one who sort of said that, species go from polyphily to paraphily. In paraphily, one of the species is monophyletic, but it is nested within the other species. And eventually you get to reciprocal monophily. And the time to monophily, what does it depend on? Let's say, barrier has just appeared. Okay? How long would it take for one of the species to become monophyletic?
It would be a function of the types of alleles and the population size.
Population size, yes. It's a function of population size. Okay? Because remember, in case of, oh God, what did I do? Oh, it goes the other way. All right. So again, going back to our example, we have two alleles, and in this particular population, there's A1, A2, and maybe A1 is going to get eventually fixed. If you have very few individuals, so A1 getting fixed would basically mean that this allele has higher, it's more likely to be sampled and will be represented in the next generation, and individuals with this allele eventually vanish, right? Now, if you have more number of individuals in the population, it'll take more number of generations for A1 to attain fixation. Okay, so here time is in terms of number of generations, right? So it's like, this room has 25 individuals and you're all randomly mating with each other, okay? And there's a new allele that pops up in one of you, okay? And that allele has to now spread, and the population is fixed for that allele. How long would it take? If it is a larger population, it will take longer, because every generation that allele has to spread, right? So that's the whole idea. So time to monophyly depends on what we call NE. What is NE? Anyone knows what NE is? Effective population size. Okay, in population genetics, we call it effective population size. Okay, so NE is effective population size, and basically what is effective population size? Do all individuals in a population reproduce and contribute alleles to the next generation? No. So the reproducing individuals in a population represent the effective population size, because only they are passing on the alleles. Right? Yeah.
One question. You said that the time it will take for one allele to fix to a population
Yeah.
is dependent on the population size.
Yeah.
Now, supposedly you have a population size which is big, but the generation gap is less.
Yeah.
So it should be dependent on the generation gap.
Yeah, generation time. Yeah. So time here in terms of number of generations, irrespective of what species, not absolute time. Absolute time, for example, in elephants will be very high, whereas in rodents will be very low, right? So here it is number of generations, time in terms of number of generations. Okay, so effective population size is what is important. If the effective population size is really small, the allele will go to fixation, reciprocal monophyly will be attained rapidly. Of course, this depends on the effective population size of both the daughter species, right? That's why we land up having this condition where one of the daughter species has a smaller effective population size, so it attains monophyly first. Okay, and because you can never have a situation in nature where both the daughter species have exactly the same number of individuals, right? So anyway, so that's a little bit of a side story here. So depending on where you're sampling, you can get gene genealogies that are different from each other. Okay. For some genes, the two populations might be monophyletic. For other genes, they might not be. Okay. All right. So this can bring about difference in gene topologies, across different genes. What are the other reasons for discordance between different genes? So we've talked about deep coalescence. We've talked about incomplete lineage sorting. So this is incomplete lineage sorting. Here the lineages have sorted according to the species. Any other thoughts on how this could occur? Oh, but before that, let's look into the complication of monophyly. So any generations? Actually, it is 4 any generations. That is the time taken for monophyly. And there's why it is 4 any. There's some mathematical simulations that people have done and have shown that. Theoretically, they have actually shown it, as well as you can write a little computer code, where you start off with one allele, new allele, and then look at many generations and see how many generations it takes. And turns out it is 4 any generations. Okay, but the interesting point here is nuclear versus mitochondrial genes. Okay, what is so special about mitochondrial gene or mitochondrial genome? Maternal inherited and it's haploid. So what does that mean in terms of effective population size? Let us assume that every individual in the population is reproducing. Okay, so that there's no confusion of young ones, old people, and all that. Okay, so let's say if there are 2 individuals in the population. So we need a male and a female, right? So 1 male and 1 female, right? So that is a male chromosome, and let us say this is female. Okay. Now both of them will have mitochondrial DNA. Okay. So let's say that's mitochondrial DNA. Will the male pass on the mitochondrial DNA? Won't. And there's only 1 here, so it's haploid, right? So already if you see the number of copies of mitochondrial DNA in this population is half that of nuclear DNA, right? Nuclear DNA, there are 4 copies, and now this is already half. Okay. Now, male does not pass it on to the next generation. Only the female passes it on to the next generation. So this particular mitochondria then gets passed on to the next generation, the female mitochondria. But male is not passing it. And this keeps happening, right? So you have to again divide it by 2 because it's only the females that are passing the mitochondrial DNA to the next generation, right? So the effective population size of the mitochondrial genome is 1/4 that of nuclear. Does that make sense? So mitochondrial DNA will attain monophyly 4 times faster than nuclear DNA. Does that make sense? Yeah. So that's why mitochondrial DNA is very useful for closely related species. When you're looking at closely related species that have diverged recently, the mitochondrial DNA will show monophyly, the nuclear DNA might not. Okay. What about Y chromosome? What is the effective population size of Y chromosome vis-a-vis autosomes? And what about X chromosome? I'm just going to draw it out and you guys better figure it out, okay? We have to do some thinking now. So that's male. So that would be XY. This would be XX. Now tell me. Which one is 3/4? X is 3/4, yeah. There are 3 copies of X compared to 4 copies of autosome. And Y chromosome? 1/4. Y chromosome is again 1/4. Okay? So, this has important implications for tree building. Mitochondrial DNA, not only does it evolve rapidly, so you have saturation problems there, and its effective population size is 1/4 that of nuclear DNA. So one has to keep all these things in mind. What is the kind of marker you're using to build your trees? So for deep diversions, mitochondrial DNA is probably not a good idea to use mitochondrial DNA. Okay? All right. Can you think of any other reason? So this is all about that. We already spoke about this. So can you think of any other reason why the... So basically the point is, nuclear versus mitochondrial trees, you can get different topologies. Don't be surprised. Right? Now, any other reason for why you might get different topologies depending on the gene you're using? Integration, yes, hybridization can do that. So there's your species phylogeny, and there's hybridization going on. So some genes have now moved between species. So for that particular gene, you're getting a different phylogeny. But for other genes, you're getting a different gene phylogeny, gene tree, right? So that is that is also something. So when you get difference in topologies, the gene topologies, one of the things you should be asking is, oh, maybe it is hybridization. Okay. Now, how do we distinguish this from rapid radiation? Because there also you might get different gene topologies. Any thoughts?
Check for distributions if they are overlapping or not. We can check for the distributions.
Distributions of what?
The species. So if they are not really having a shared region, then their chances of overlapping would be gene flow would be very less.
Okay. So that is, current gene flow. So if there's no overlap. That is. But what happens in case of a rapid radiation? Yeah, tell me.
You might get mitochondrial DNA from B and C.
Yeah, so mitonuclear discordance is one of the sort of indications of hybridization. But what if 2 nuclear genes give you different trees? This could be just because of deep coalescence, or it could also be hybridization. So how do you distinguish? What happens in case of rapid radiation? In terms of the speciation events? They are very close to each other, the 2 speciation events, right? So what is going to happen in case of rapid radiation? When you look at the gene genealogies or the gene trees, what's that? Yeah, branch lengths will be very small. So it will be like that. Right? Whereas hybridization case, the branch lengths need not be very small. but the topologies will be very different. Even here, the two topologies will be very different. This will be like this, but this branch length will be very short, whereas here the two topologies will be different, but this time period need not be very different, right? So there are ways to like figure that out just by looking at the tree, what's really going on, right? So it's very important to try and understand what kind of markers you're using, what might have been the ancestral population size. I mean, all these are very important things one needs to keep in mind while trying to interpret the tree, right? Okay, hybridization. So someone also said selection, okay? Selection, as I said, if there's one particular gene which is under selection, positive selection. And remember, positive selection is when non-synonymous substitutions outnumber synonymous substitutions, right? So if the genes are under positive selection, you should not use them to build phylogenies. Ideally, one should use neutral markers, but neutral markers are difficult to come by because most of the sequencing that has been done in the last 30, 40 years has been some gene or the other, right? So all the information in terms of primers, sequences are there for the genes. Now, as long as the genes are under negative selection, it's okay. You can use them. Because any change that you observe between those genes are all neutral changes. They are not resulting in change in amino acid, right? The problem arises if you use genes that are under positive selection. And there have been many instances where people are beginning to realize they have to look at this more closely, right? So here's a case where this particular gene has been recruited for the same function in unrelated species A and D. So this is basically, it results in conversions. So this is conversions at the molecular level, right? And I talked about the gene lysozyme that has been recruited for the same function in colobine monkeys as well as in some lineages of ungulates, right? And so you have cow and langur branching together because they share the same amino acid replacement. The sequences are pretty much identical, right? So this can also result in some genes giving you one topology while other genes giving you other topology. Okay, so this is the actual tree topology. Most genes will give you that, but the genes that are under positive selection will give you something like this, right? Okay, any other reason one can think of why gene trees and species trees can be different? I think we have covered most of them. Yeah. Right? So hybridization, a very important reason. You can also have horizontal gene transfer in many of the bacteria. Right? Horizontal gene transfer is somewhat like hybridization. Unrelated species would branch together because there has been a horizontal gene transfer. Right? So for that particular marker, you'll get a completely bizarre phylogeny. In fact, the mitochondria in eukaryotes, what is the origin of that mitochondria? Huh? Louder.
It's an endosymbiotic.
Yeah, it's basically an outcome of an endosymbiotic event. But where did it come from? Bacteria. Alpha proteobacteria. Right. So if you take a bacterial build of bacterial phylogeny and throw in the mitochondrial sequence of eukaryotes in it, then that will go and branch where there's alpha proteobacteria. right? So it is horizontal gene transfer. So we talked about retention of ancestral polymorphism in case of recently diverged species. Then this is deep coalescence. Gene divergence precedes species divergence. These two are pretty much the same, but the only difference here is that here enough synapomorphies have not accumulated and there has been reversal, right? And then the biological property of the marker is also important. Effective population size, the sex ratio. Sex ratio can make a huge difference, and also the mating system. Right, in groups that exhibit very high variance in reproductive success, say male reproductive success, very high variance. What do I mean by that? Few males do all the reproduction. Most males are not able to reproduce, right? So only those few males will now pass on the Y chromosome to the next generation. So now the effective population size of Y chromosome goes down even further, right? And that's going to have implications on how populations are related and things like that. Okay, so just bear all this in mind when you are doing your analysis. What kind of marker you are using, what kind of inheritance it exhibits, how rapidly it evolves, and all that, because that's very important. And if you see discordance between two genes, don't be alarmed. It could be any of these reasons. Ideally, we don't expect to see a discordance. But if you do see it, it's not a huge surprise. Okay? All right, any questions so far?
Basically, we should select a gene with a bigger effective population size.
No, if you are using multiple markers, they should all have the same effective population size. If you are using mitochondrial gene and nuclear gene, just bear in mind that these two have very different effective population size. You might actually get the same tree topology, but just bear in mind that they have different effective population size, and you might get a different tree topology.
What kind of context?
Yeah, you can. Yeah, we always. It's only when they are different. If you get the same topology, you're fine. I mean, just go ahead with your analysis, right? It's only when they are strikingly different. Then you go to this presentation and look at, what could be the reason, right? One of them is mitochondrial. Yeah, not a big surprise. Mitochondrial, the other issue that crops up when you're looking at deep divergence, you should not use mitochondrial DNA for deep divergence. Looking at higher level phylogenies, because of the problem of saturation. Mitochondrial DNA evolves at a rate 8 to 10 times faster, in some cases 30 times faster than nuclear genes. So you'll have huge saturation problem with mitochondrial DNA, right? Okay, any questions? All right, before I move on to the next one, I'm sorry, I got to quickly run to the restroom. I'll be back in 2 minutes.
Yeah. No, it is on.